Synapsis is the process of homologous pairs of chromosomes coming together during prophase 1 of meiosis in order to exchange genetic information between the homologs. In order to maximize the pairings of homologs, we want their regions to match up. Individual 1 has these chromosomes. In order to match up the regions of these homologs, we can invert chromosome 2 like this. The pairs now match up better than before. However, these regions are still not matching up. This is due to the fact that the region I.JK experiences a pericentric inversion. In order to fix this issue and to line up all of the regions correctly, we must form an inversion loop. Here is our inversion loop. As we can see, individual 1 has a crossover occur between the J and K regions of their chromosomes. The crossover only affects the inner two chromatids, whereas the outer two chromatids are not affected by the crossover. In order to predict the 4 chromatids that these chromosomes will produce, we must simply follow the lines of our inversion loop. Following this outer line, we get a chromatid with this sequence. Following this inner line, we get a chromatid with this sequence. Following this outer line, we get a chromatid with this sequence. Following this line, we get a chromatid with this sequence. As we can see, these two chromatids produce viable non-recombinant gametes with the same sequences as their parent chromosomes. These two chromatids, however, produce non-viable recombinant gametes with sequences that are either missing regions and/or repeating other regions. Individual 2 has these chromosomes. In order to line up the regions of these two chromosomes, we can invert chromosome 2 like this. As we can see, all of the regions line up perfectly. This is because these chromosomes are homozygous. Due to the fact that there are no inversions, we do not have to form an inversion loop in order to make our crosses. Individual 2 also has a crossover between the J and K regions, as we can see here. Just like in the inversion loop, only the inner chromatids are affected by this crossover. In order to predict the 4 chromatids that are produced, we must follow the lines once again. Following these lines gives us the following chromatids. As we can see, each chromatid produces a viable non-recombinant gamete that has the same sequence as the parent chromosomes. None of the sequences are missing any regions or repeating any regions. Based on the gametes from individual 1 and individual 2, we can see that individual 1 gives us 2 viable gametes, whereas individual 2 gives us 4 viable gametes. This means that individual 2 has a higher fitness than individual 1, and we expect their offspring to have a better rate of survival.